Testing 1, 2. Okay. Okay, folks, let's get started. Let's see. So, since we last met, your screen's fixed, so we're good. Although this is kind of not very bright, but anyway, welcome back. Glad you all came back. This is your first day. And you don't have Blackboard access. If you do already, then you got everything you need. But if you do not yet, it may take a couple of days in some cases. Send you. Oh, a couple things. So I've already uploaded the Monday's Blackboard. Those of you that had a clicker and used it. And I registered whatever points were, not I uploaded whatever points I received. So if you look on Blackboard, you'll see if you use a functioning working clicker, you should see points, and you should see a total points thing for you. Again, that's all going to go away in a half. So remember, next Wednesday, week from today, they start reading. So and at that time, I'll just delete the data. Data is to, to ask questions in class and back in that kind of. But they're not counting for points yet. But so in the meantime, make sure the purpose really is to make sure your clicker's working, that you're getting your points, that you're seeing what you're supposed to see. So that's so look at that. Do look and see. If you don't have a clicker yet, you're not going to see anything. But if you do, hopefully today, if you don't have one, Monday, I went today or next Monday with holiday. That's right, we're not going to be here next Monday, so I won't see you for another week. So, just check Blackboard, make sure you get your points. They're registering, and everything's good there. Okay. Again, if you have issues with it, CB202, and help fix whatever issues you got. Let's see, what else? There was, but I can't remember. Is there any questions or anything? Okay. So we are going to do a bunch of clicker questions today again. So it's always going to be channel 41 all the time, 41. Okay. And I usually we'll start here in a couple slides with a question to get the old brain bone working. Okay. Thinking about micro. So just kind of a recap. So this is kind of where we got the very beginnings of chapter 1. So what we're doing obviously for the rest of the day, and I think left on for next Wednesday, is just really just an overview. This is chapter 2. What are microbes? Here's what they are. What are the basic features? How do you define them? Where do they come from? What do they do? All these kind of things. And so not going into super detail on these, because that's what the rest of the semester is about, but basically it's kind of giving you an overview. This is what we're dealing with, okay, this semester. So we really ended here with the good and the bad, right? So we're talking about microbes, or good guys or bad guys, but if there's one thing to take away, that I want you to take away from this course, is that at the end, you know what? Most of the stuff I hear about in the news and whatnot, it's all disease-causing microbes, that, most of these things we need, we rely on. We must, if we didn't have these certain microbiological activities, particularly in this category of ecology and here symbioses, we literally would not exist on this planet for the things they provide, okay? So directly and indirectly to us. So the idea that they are good. Most of these things are good. We need them. We must have them, despite the fact that we do have to deal with infectious diseases. But regardless, okay. So again, good, bad, okay, and then that's basically where we So we'll start with we are starting with a clear question. Okay, so here we go. So your choices answered, so you want to basically pick among answers G through J, but A through F are what you're evaluating, right? You're evaluating A through F. One are ones of these. Okay. So A, B, C, D, E, F. Some of these things, all these things, are stuff we're going to today. Okay, let's just see if anybody's prepared for lecture. Okay, you can talk amongst yourself. Okay, if you find that helpful. Okay, don't be shy. All right. Oh, my main thing I do. This is one thing you have to constantly remind me of. Is open the stupid poll, idiot. Because I always forget to do that. Let me get my. There we go. Okay. You can remind me just on there. Remind me. Okay. All right. Okay, I'm going to let it go. So 168 looks like our head count. 69, 70. Whoop. Okay, here we go. 1, 2. Okay, G. All right. Okay, so G 71%. Remember that. All right, so let's go through the. So G, 3 are true. Okay. What's one that's true? B. B is yes, that's true. So B is a true statement. C like a consensus. C is true. And anything else? D is a dog. Yes. Those are the true statements. But 3%? That's pretty good. Okay. That's where you really know this stuff. That's good. I'm impressed. So what do we call process B as in boy? No. Process B is not nitrogen fixation. Nitrogen fixation uses N2. It's the process called You're familiar with what you all learned in intro bio was aerobic respiration, right? This is anaerobic respiration. Okay. What's an example of a molecule C that a prokaryote can use in that way? An inorganic source. An example of it. What do you know? Yeah, absolutely. That's what they find there and use. Methane, possibly. H2S, yes. Iron sulfide. It's going to be what we call reduced organic compounds. So we're talking about oxidation reduction in 4 weeks, but a reduced compounds. So reduced means they're heavy in electrons. So like H2S, as you said, hydrogen gas. So nitrogen compounds like ammonia as well. Iron can be used as energy sources. But only bacteria and archaea that can do these kind of things. B certainly is true. That's a cyanobacteria type of bacteria, photosynthesized basically the same way as a plant does, chlorophyll, etc. But they are the ones that put O2 in our atmosphere. So the ones that are false here, just to point out, that's false because you want to change millions to billions. Add 3 more zeros there, okay, to make that a true statement. It's false as written. F wasn't the one to connect the, although we'll talk about that. It was Koch who did that germ theory of disease. And liver cells, that's kind of A is kind of the first thing we're going to touch on, that kind of idea of defining a microbe. Any questions about that? Okay? So let's look at that, basically defining a microbe. So it's and there can be, it's not often the case in biology, I'm sure you've found out already, that not everything necessarily fits is a black or white answer, right? It's always only this or it's always only that, okay? But there are going to be some overlapping areas, some kind of what you might call gray areas, what seem to be contradictions, but those are just things you have to be aware of, but know that maybe just follow a certain trend, but you can have some of these oddballs here and there. And the same is true for defining a microbe, but you know, the most basic thing, what do you need, how do you define it? Well, you need a microscope to see it, right? Super tiny, you can't see it with a naked eye. That's pretty much one of the basic criteria, but then there's lots of things that could fit into that. But for our purposes, basically talking about microbes. It's a microbe is something you can throw out in the environment. It can survive on its own. It can replicate. It can use nutrients and produce energy. It can replicate, etc. But you can't have very tiny metazoans. We are metazoans. Okay, so metazoans are multicellular types. Okay, but even those can be rather tiny. And in some cases, you may need to use a microscope. But generally, that term microbe is reserved for microorganisms that can be have certain shapes and arrangements. Think of like streptococci or cocci in chains. Okay, they may have some of these kinds of arrangements, but they typically are kind of single units, or they can have arrangements. They are basically cells for the most part, okay? But we do have, again, some of these exceptions like viruses, okay? Viruses absolutely need a microscope, an electron microscope to see them, okay? But they have kind of this, you know, are they alive or are they not alive, okay? They're alive in the context of being, when they're inside of a host cell, they're alive because they're replicating themselves and making copies of themselves. But when they're outside the host, you don't really know they're not really doing anything. They're kind of in this suspended animation kind of a state, right? I can almost be pretty certain there's probably some viruses on that handlebar right there, but they're not going to do anything until they can infect a host cell and then replicate themselves. So while they're there, are they really alive? When they're inside of a cell, definitely. Okay, so they kind of have straddle those two kind of worlds, if you will. Okay. Now, I mentioned a little bit about sizes. Okay, and the scale of sizes. So viruses, of course, the smallest. So we're talking in terms of tenths or hundreds or thousands of micrometers. Okay, the prokaryotes, bacteria, archaea, we're kind of in that 1 to 10 micron range. But eukaryotic microbes, another 10x increase, 10 to 20x increase in size beyond that. So, and this kind of gives you a scale of basically cellular and particles and molecular particles in relation to cell sizes, and of course super huge types of cells. Okay, eggs, right? Chicken egg. Okay, so I did this question: what determines the minimum size of a cell? So how small can a cell get? So here's our other question. Okay, and that's asked. This is just be a quickie one, just a yes or no. Hypothetically, could a cell exist that is that small, one nanometer in size, 0.001 micron? Okay, super tiny. Okay. Didn't want that. Sorry about that. Didn't try it. I will do some of this stupid stuff during the semester, so get used to it. Okay. Okay. You stragglers out there. 152. All right, I'm going to let it go. Okay. Beep. All right, so majority says A. Who's somebody that said no? Who said no? Why do you say no?
It needs so much room to fit everything inside of it.
Exactly. Right. That's why that's the correct answer. It could not be super tiny. So if you look at the diagram here, you can see the small stuff down here, right? So things like small molecules, lipids, okay, are really in this very small range. But if you start combining lots of ribosomes are in a cell are probably one of the most abundant molecules. The ribosomes are kind of big, okay? And when you start having hundreds or thousands of them, it's going to be hard to fit that in a 1 nanometer cell, okay? So, the chromosome is big, right? So you're limited by the stuff that cells need, which is why, okay, that viruses have kind of this, that's why they can be so small, right? Because a virus doesn't do a lot of stuff on its own, right? It needs a host, okay? So that's why viruses can be super tiny, because they don't carry a lot of stuff, right? They have a chromosome, which is very small compared to other cellular-based life, and proteins, about it, inside them, so they can have very small sizes for that reason. Okay. All right. So kind of, I don't know if gray areas is the right term, but let me get this thing out the way. But there are some kind of contradictions in some cases, but certainly tissues, right? Your own tissues, your skin cells, your brain cells, what have you. Yes, you need, we look at those things with a microscope, but we don't call them technically microbes because they really wouldn't exist without being part of a larger tissue, organ, what have you. Your cells are very well protected slash taken care of in your body. Your body provides all the right conditions and temperature and etc. Any of those cells making up your tissues would not survive on its own out in the environment. Okay. The viruses, we said, yes, I do consider these things microbes. Okay. Although they're not cells, so they're kind of that gray area of is it alive or not. But generally, they are considered microbes, microanimals. Right? So those metazoans, right? Multicellular types. There's a water bear, they call it, that can be super tiny. Maybe need a microscope to see it, but we don't generally refer those as microbes. Yeah, I think so. Some differentiated tissue. Yes, they do. And this guy. Now, this guy I do consider a microbe because it is it's a archaea, I believe. But it happens to be big when this vesicle here fills up with a gas. Okay, so this is a fruit fly for comparison. Fruit fly head. It's that structure that fills up with gas, and so H2S. We talked about about anaerobic respiration just briefly. That these guys use H2S as a energy source, but they use nitrates as their molecule to, quote, breathe with, right? Like we use oxygen at the end of our electron transport chain and convert that to water. These guys will take nitrate and convert it to nitrite, and then use H2S as energy. So they have this unique metabolism, but what this does for them by filling this sac with NO3, it's kind of like their own personal scuba tank, right? So they can have a really supply of if you will, and allows them to survive in their environment, right? So if they may be in an area where there's no or very little of this, but they have it with them in that big sac, they can survive in those areas. Okay, but that thing can really expand so big that it becomes visible. Okay. Okay, so microbial community. So these are not microbes. I mean, they're made up of they're made up of individual microbes, of course, right? A colony on a plate, right? An individual bacterial cell would not be visible to the naked eye, but if you then combine it with 1 million, 10 million of its neighbors, you'll see a visible colony. Okay. So microbial communities, so biofilms are very important, both environmentally, particularly from a medical standpoint. Biofilms are all about surfaces, okay, and about microbes sticking to a surface and then producing kind of a glue, if you will, to keep everybody stuck together. And they typically will reside in areas that are nutrient rich, okay, because you got to feed everybody in that biofilm. So it's very common to see them in different types of pipes, for example, sewer pipes, because you'll have organic heavy material, liquid flowing through, and that can feed everybody. Then they can begin to stick there. You may even say that if you haven't cleaned your shower curtain in a while, you get a film on there as well, and that can be a biofilm. Your teeth, right? That kind of sticky feeling that you wake up with in your mouth, that's biofilms on your teeth, right? So, and think of, they have a nutrient-rich solution they're getting fed, right? Food going through your mouth, okay? So, lots of places where biofilms can form, okay? And there's different phenomenon that we'll see during the semester that kind of drives a lot of this microbial behavior, if you will, and one of the biggest drivers for that is nutrients. Is it rich in nutrients, or is it nutrient limited? And that can trigger different types of behaviors. Nutrient rich, biofilms can form. nutrient poor, then you can see things like spores forming and stuff like this. So we'll see different examples during the semester where different nutrient conditions can bring about very different types of microbial behaviors depending on what you're dealing with. Okay. So where are they found? Okay. Well, the word you always see associated with this question is that. Right? Everywhere, ubiquitous. They're found all over the place. Okay? Extremes of temperature, cold, hot, on your body, in your body. Okay? A handful of soil, a tablespoon full of soil has bazillions of microorganisms. In the upper reaches of the atmosphere. Okay? Some even think that maybe they may play a role in some cases in the formation of rain droplets and other weather phenomena. Okay? Here is the Red Sea. This is like, have like salt concentrations of like 20, 25%, right? So imagine, what in an environment like that, what's that physical property that the cells are going to be fighting with to live in that environment, right? Because what's a cell going to be losing when it's in a super salty solution? Water, right? So there's adaptations right there. All the microbes, of course, have various adaptations to survive, and not just survive, but thrive in these environments. So the microbes you see here aren't just getting by and eking out a living. I mean, they need that 25% salt to grow very quickly. Okay, so they're not just surviving; they are, in many cases, thriving in these environments. Okay, they're built for it. Okay, so here we have a picture of Mars, and so I have a question thrown in. When space scientists see evidence for life on Mars, why do you think they find microbes rather than creatures like the alien monsters often depicted in science fiction? E.T., this is very typical, right? The oblong head with the big eyeballs, you always see that as an alien, okay? But why do they always point to microbes? Why is that what scientists look for typically? Anybody?
To survive in extreme situations.
Yes. Yeah, so it really goes to that. So if you look back at this diagram here, so Mars environment is going to be very cold, okay? So something like this. But it's precisely for that reason, because what do we find in extreme environments on Earth, right? Whether it's extreme cold, salty conditions, very hot, what have you, is microbes. You don't see metazoans like us, right? Thriving in areas of microbes. Okay, and so expect to find it's going to be something like that. Okay. I certainly believe that. I don't expect to see something like this running around up there. So discovery, just kind of quickly here. I think we all heard this story. Van Leeuwenhoek, of course, we all, I think, are aware of. The thing that differentiates these two, Hook and Van Leeuwenhoek, is, and they were contemporaries, 1600s, is Hook was number one in terms of using a microscope and looking at stuff with a microscope, but he looked at various types of tissues, whether plant tissues, animal tissues. He looked at things like fleas and butterfly wings and stuff like that, right? Not really the first to look at microorganisms. His microscope was actually rather puny in terms of magnification. 30x is not, you're not going to see bacteria with 30x, okay? But Van Leeuwenhoek had lenses that, and I don't know nothing about grinding lenses and what that all entails, but apparently he was some kind of a Rain Man when it came to lenses, grinding lenses, okay? Some kind of a genius apparently, but he was also very secretive. He didn't want to tell anybody how he did it, okay? But he published all of his findings, and you look at the drawings from way back then, and you can certainly identify that these are certainly protozoans here. These are bacteria. So, and I'm thinking his magnification they thought was around 200 to maybe 300x. So you can see some of your larger prokaryotes with that fairly well. So anyway, so Van Leeuwenhoek and his animalcules, okay? Now, of course, we're talking 1600s, okay? There's all kinds of, what comes about then is where do these things come from? They're invisible to our eyes, and now we're seeing this stuff. What the heck is it, okay? And so, and where do they come from? So now you get all these kind of crazy ideas, which have already been around for a while. This is actually something that Van Leeuwenhoek thought he saw, because he examined sperm, okay? And he thought he saw this in the head of a sperm, okay? And that's, you look closely enough, right? It's actually a fully formed human, okay? So I don't know what he was smoking that day, but this was part of a movement because I think they were called, aptly enough, spermists, okay? And they believed that humans reproduced, babies are born, and the fully formed fetus was in the male sperm, okay? Woman didn't have a role other than like being an oven, right? 350 for 9 months, and out pops the baby. All right, that's her sole contribution. Okay, so that idea certainly wouldn't fly today, but obviously it's ridiculous. So, but you know, that's, I just bring that up to illustrate that there are this kind of in the environment where spontaneous generation comes in. Okay, that life can just spring forth from spontaneously from nothing. Okay. You had to have things like, the one thing you did have to have was air. They really believed you had to have air was part of the process. You had to have that in there to get these inanimate objects together to make a life. And so this is a picture that shows, you know, if you have, you know, you leave grain, like if you have horses or cattle or something, and you're out in your barn, you have your grain, right, for your horses. That if you don't tidy it up and leave the lid off or whatever, and it can get wet and moldy, stuff will grow, right? Fungus and get rot, right? And you likely will see rats and mice running around eating it, right? But don't make the connection, okay? You leave that the grain can bring forth mice and rats and stuff, right? It's not, spontaneous generation is as we all know, ridiculous, okay? So, but people believe that for decades, okay, if not hundreds of years. And so of course there were some people around that goes, okay, was this really true or is this just, you know, made up, okay? And so then came a series of experiments during this time, probably around the 17 through 1800s, trying to, you know, is this real or not real? And so Spallanzani had one of these experiments where he looked at, so the idea here is you have broth, right? And which basically be some kind of a beef soup broth, okay? And you heat it, right? To presumably kill anything in there. Leave the flask open, and you wait a while, and of course stuff will grow in it. We all know that, okay? And then, but you know, spontaneous generation, people were saying, this is life springing forth from non-life. There's your evidence, okay? So Spallanzani goes, okay, I'll just seal the flask, okay? Boil it, seal it, and nothing ever grows unless I open the flask, okay? And he goes, aha, see? And they go, well, this is not, that doesn't count, all right? That's no good, all right? So the people that believed abiogenesis, another name for spontaneous generation, said, no good, because you're not letting air in there, all right? That's the vital ingredient, right? You need to have air in there, okay? So then this brings in Pasteur, okay? So Pasteur was a chemist by profession, but if you've taken chem, he's the one that kind of figured out the left-handedness, the chirality of molecules, the left and right-handedness of molecules. He's the one that figured that out, among other contributions. But even though he's a chemist, he was famous for the stuff he did in microbiology. So, and among those was really figuring out the metabolic capabilities of microbes. Okay, we'll make our way back to spontaneous generation here in a second. So he, the French loved their wine. Okay, and so there was a period where the wine being made had this weird taste to it. Okay, basically tasted kind of like vinegar. And they would make batches where this would happen. And so they called Pasteur in, who they know was a famous chemist, of course, to figure this out. And so he looked at the process, and so, you know, I just very simplistically put it up here, right? Grapes, of course, are what the sources of the sugar that gets fermented, okay? So in the absence of air, which we call anaerobic, we have the fermentation of sugar to alcohol, okay? Which occurs during wine production, okay? Simple enough. And so when he looks at that then more closely, seeing in the flask that there's cells attributed to this transformation making this alcohol, right? That we see the production of cells along the way, yeast, right? And the yeast are basically coming from the grapes themselves on the surface of the grapes, okay? And as the yeast proliferate and get more plentiful, which you can see by tracking from samples using a microscope, that there's more and more yeast, and that equates to more and more alcohol. Right? So now we're seeing the link between, cells are causing this metabolism. It's not just a strictly chemical, biological reaction. It's a biological reaction occurring because of the growth of microbes. Okay, and they're transforming the sugar, okay, the grapes, to alcohol, okay? And so he goes, okay, well, that's what's going on, but then what's causing the deviation where the wine doesn't taste right? What's that all about, okay? Well, of course, not only will yeast do this, but so too can bacterial contaminants, okay? So bacteria can take that and make things like acetic acid, among other things, right? There's various acetic acid, propionic acid, butyric acid, generally formic acid. So generally like your C1, C2, C3, C4 acids and alcohols, okay? So this is where he found that he could see that, if a certain bacteria contaminates, then that will make butyric acid. If it's a different one, it'll make acetic acid. So he basically differentiated and associated various bacterial types to different fermentation products, we call it, in this process. And so this is where the germ theory of fermentation comes out of this, okay? So number one, all these reactions going on are due to microbes. It's not just a strictly chemical reaction, okay? It's cell-based chemical reactions, if you will, okay? Again, when you go from beginning to end, you'll find that you have very few cells here, but lots of cells here and lots of product, right? So it's cells that are creating this. The end products, you can often, and you can do this today as well. If you carry on a fermentation, those that have done this a lot, you can smell the test tube of a flask after the fermentation is done and kind of go, oh yeah, that's kind of this alcohol or that acid, and you can kind of identify it by that. And so this was huge, right? So now we have the transformation of organic materials occurring through the action of microorganisms and their metabolism, okay? And so Pasteur then kind of sets his sights on this spontaneous generation argument, okay? And he goes, okay, I'm going to ingeniously design this very simple experiment where he fulfills the requirements of the spontaneous generation people saying, got to have air in this system, right, or it doesn't work. So he makes a flask here that looks fairly normal, right, with broth in it, but he has this real long swan neck on it, okay? So, which is exposed to air, okay? Air contaminate something because they're flying on dust particles, okay? Or settling on dust particles, and that's how they get into and contaminate. So with this system, air can come in, but the dust settles just by gravity in the crook of the neck right here, okay, and never gets into here unless he were to tip the flask so the liquid then goes down into here, or breaks it off right there, exposing it directly to the air. In that case, it will get contaminated, okay? So he basically shows that with this, and they claim that there are still flasks like this sitting in his lab that have never been contaminated in 150 years or whatever it is, okay? But regardless, so he says to them, well, here's air, but I'm not getting growth, right? No growth, and of course, by tipping the medium into here, cells appear. Look under the microscope, and cells begin to appear in here if he does that or exposes it directly to air. So in other words, we're only getting life if we add life in the first place, okay? And so it wasn't long before the spontaneous generation people gave up, okay? But there is one side note here, and that is Tyndall, okay? So he repeated Pasteur's experiments, whereas Pasteur didn't see the phenomenon, he did. And the phenomenon was that sometimes when he did this, in some cases he would get growth even though the whole system was intact and he didn't carry over any liquid over to here. He would get growth, okay? This is the discovery of endospore formers, okay? So you've heard of spores in various contexts. Plants can have spores. Fungi can have spores. There's even other bacterial types that can produce spores as well. But it's the when we put this in front of it, endospore, that's a very unique type of spore, very resistant, okay? They've literally isolated endospores from material that's been dated back 250 million years ago and have revived those spores, and they've germinated and grew cells. So they're very resistant. And these are the two genera that are of those types. There's nothing else other than these two groups. And this group, Clostridium, is one that there's a lot of pathogens in that group, and toxin formers, tetanus, for example, botulism, gangrene, if you've heard of that. All are members of this group, and all of them, a lot of them, most of them, I think, all produce various toxins. Okay, Bacillus is the most dangerous one in that. They're mostly benign types in Bacillus, but the one you've likely heard of is anthrax. Okay, that's in that group. But the other ones are pretty much aren't human pathogens. But both of those types are all basically soil organisms in the soil, but very resistant. So what happened, what he did was, he would let's draw a crude flask here. He'd have a flask, and he'd have one of these swan necks on it, okay, and very ugly swan neck, excuse me, and he would get growth, okay. And so he would look in and see these forms that had kind of this encased in, kind of looked very different. So, and you see an endospore, it has a very unique depiction of it. So he'd get growth, and then what he would do is he would sterilize, he'd boil it, okay? And so what would happen is, as the boiled broth, okay, would initiate spore formation, right? And as it went down to temperature and cooled off, then some of those spores would germinate and grow, okay? And so then if he boiled it again, he would kill off those types, okay? So, but then there would still be a handful of spores left, and they would grow again, okay? So then you have to keep repeating the cycle, right? So at each time, some of the spores would germinate as the temperature came back down, and you kill them at boiling, and then if you keep doing that a few cycles, eventually you'll get a sterile broth, and you would after maybe 3 or 4 cycles. So, but again, it's attributed to these, and frankly, that's why we have an autoclave over in STL to make media for lab, because we need the autoclave to kill endospore formers, basically. Okay, that's what will kill them. Okay, or extended boiling, but that's kind of impractical for making media. So, so there's this kind of important discovery there with that. Okay, now, okay, so take a breath and look at this question. Okay. Because we're going to segue into the next part. So how does the germ theory of fermentation relate to germ theory of disease? Okay, let's kind of think about Pasteur and his work with germ theory of fermentation. Okay, so do they both explain the infectious disease state? They both require animal models. They both relate to changes in organic matter due to the action of microbes. They are not related at all. Okay. Oh. Didn't say, hey, idiot, turn the polling on. There we go. I think you're going to figure this one out pretty easy. Oh, not what I wanted. That one. Okay, so, okay, so let me show you with this. Okay, so germ theory of fermentation. So attributing microbes to production of transforming, organic material to end products. Germ theory of disease. Right, healthy human. Sick human, excuse the gross picture, okay? So the connection, right, is exactly what you chose in the previous question, right? They relate to changes in organic matter, right? So, I mean, these are certainly organic beings, right? We're organic matter as well. So the idea was, okay, well, Pasteur showed, that microbes can transform organic matter in a flask, right? Sugaring grapes to alcohol. Well, then maybe that's, analogous to what happens when a person's sick, right? The transformations are occurring to make this body unwell, okay? And then that's kind of where Cope takes over and kind of really nails that down, okay? And so this is the germ theory of disease, all right? And so what we call a chain of infection, establishing that microbe X causes disease Y, okay? And doing it. Obviously, what we're talking about here is infectious diseases. We're not talking about things like diabetes or disease or cancer, right? We're talking about infectious diseases, which there is a microbe attributed to it, okay? So etiology is kind of that fancy word for cause of disease. And so Cope kind of did this in two parts, if you will. And he was kind of lucky out of the gate in establishing cause and effect by looking at anthrax. So anthrax is at the time he was looking at it, was kind of, he was a doctor out in the country. So of course he could see the effects of anthrax on the livestock around him. So there was like this outbreak of anthrax, cattle were dying, and it becomes what we call a septicemia sets in the cow, where the bacteria basically flowing through the blood, okay? And so all he really did was looked at, okay, here's diseased cows, here's healthy cows, he takes blood samples, looks under the microscope, and he goes, I'm only seeing this, all right? That's what they look like, Bacillus anthracis looks like. I'm only seeing this in the blood of sick cows, not healthy cows. And now it begins to make the link, right? So that's, and if he took blood samples containing this and put it in a healthy animal, the animal would get sick. So this is where we're fairly easy to establish it when you can find out the cause of disease by looking at a blood sample, okay? You don't have to do a lot of work. But there are, of course, those kinds of diseases. That's fairly simple, but there's going to take more work, trying to figure out the cause of tuberculosis, okay? Because now it involves, you got to culture it. You got to isolate and culture it. And so this actually was huge, figuring out these techniques and ways to do this, because once this was figured out, this had application across the board in all areas of microbiology, okay? You want to study a bacterium in the soil? Now you have a way to do it. Take samples, grow them up, plate them out, and then study them, okay? So that's going to apply to all areas of microbiology. And so this is like the 1870s. So the 70s, the 80s, and 90s, they call the golden age of microbiology because of this, and allowed scientists to look at microbes of all types and have ways to study them, okay? And so the development, and you'll learn this in lab starting week after next, but developing aseptic pure culture. So aseptic, doing work without introduction of contamination, okay? Why do we have to worry about that? So remember Pasteur, right? Then the flask, you expose it to air, stuff comes in and contaminates, right? So microbes are everywhere, right? So if you want to study a certain microbe, you want to do so in a pure way. You're only looking at it and nothing else. So you have to do things in a way to minimize contamination from the air and wherever else, okay? So that's what that all refers to, and there's different techniques that allow you to do that, okay? And of course, the two basic types, liquid growth in liquid medium, growth in solid medium. Two different purposes for those things, but essential purposes. If you want to grow up lots of cells, okay, for purposes of isolating DNA, isolating proteins, what have you, you use liquid, okay? It can be a part of attaining a pure culture, but only a part. It's not going to be, because you can have a mixture of cells in here, and you can look under a microscope, take a sample, and see that, okay? You may see different types swimming around or what have you, but you cannot manipulate, right? You can't, you need to get something you can work with, okay, on a plate and distinguish them, right? So here's a mixed culture, right? See yellow types and more opaque types, right? So you take it out of here, put it onto here, so you can see visible colonies, and then you can manipulate. You can take this guy and do something, and take him, put it on another plate, pure culture, right? Take this guy here, pure culture. So now you can get pure cultures of different types only by going through this step, okay? Because you can't do that with just a liquid culture unless you have a pair of tweezers that you can pull out cells out, which of course you can't, right? That's why solid media is important in that process. So again, so developing these techniques was big in terms of all of microbiology, really. Now you can study these things. I'll skip that question. I'll do that one next time. So here's a question that relates to the next thing we'll talk about. Okay. So this is about which is a false statement among A to D. So and again to the pathogens and what they can do or can't do. Okay. So look at each of those four. gosh, that's... Okay. Okay. All right, folks, let's make a choice. And okay. So E and F. Okay. Let's see here. All right, so which is the false statement? Healthy persons can carry pathogens without becoming diseased. Is that true?
Yes.
Okay, so what's an example of that?
So for instance, some people with HIV can go for years without showing any symptoms, while other people like die in like 2 months.
Okay, are the persons that have the HIV but not showing symptoms, will they eventually succumb to the disease?
Eventually.
Okay.
It can take up to a decade in some
Okay.
cases.
I'm going to say that that's not the best example. Okay. Does anybody have another example? No, I don't think. No, that's a tick-borne disease. Yeah.
What about latent tuberculosis?
Latent what?
Latent TB?
No, because I'm going to say that that person can still succumb to tuberculosis. I'm not aware of healthy people with TB. I think that's kind of a gray area. I'm looking for a little bit better example. A what? No, it's going to be, we're talking about people that have it are what we call carriers, and they never show any symptoms of disease at all, never succumb to the disease, but are the ones that hand it off to other people. So certain disease outbreaks that occur, yeah. That's not infectious disease. Yes. It could, yeah. I think that may fit. All right. Do you have an example?
Yeah, I was thinking there is some bacteria that are they're inside some animals, but they don't kill them. But when
Yeah.
that animal comes in contact with a human or some other animal,
Right.
the bacteria go into that animal and actually cause the disease.
Yeah, that can happen. Maybe along those lines, like E. coli 0157, where it's the Chipotle E. coli, the lettuce, that one. That one's actually in cattle, and cattle show no signs don't come down with the disease, but they spread that through their feces into the crops that are maybe nearby, and that's how we end up with contaminated lettuce. Yeah. Which one? Yeah, herpes virus, yeah. The one I'm really thinking about, because you all got vaccinated for this before coming to school. Meningitis. Is that what you were going to say? Okay. Meningitis. I don't want to, because you said tuberculosis. Somebody else said HIV. Typically, when you look those diseases up in the textbook, TB is generally it's an airborne infection. There's a term called reservoir, a reservoir of infection. That's the source of the pathogen. If you want to find the pathogen, where would you go? You look at the reservoir, where it naturally resides. Of course, it has to get from that reservoir to you, and those are different modes of transmission. Okay, through the air, through water, through food, eating contaminated food. TB is generally not. Yeah, anybody who's sick with a disease can technically pass it to somebody else, but we're talking about a carrier is unique in that they show no symptoms of disease and don't have the disease. They just are carrying the organism, okay? And meningitis is one of those, like, I think 40%, could be even higher, of just the normal healthy population carries it in their throat, okay? But when it passes from them to a susceptible host, then that's when outbreaks occur. And so meningitis commonly occurs in, that's why you got vaccinated, because many of you live in dorms in closed quarters, and that's where meningitis can spread. So a healthy person just carries it in his throat, but then a susceptible person picks it up, then spreads it to X, Y, and Z. Right? Yeah?
What about sickle cell trait, just having the trait but not the actual?
That again, that's going to be, that's passed on through, that's a genetic mutation. So you're going to pass that on that way. So that really doesn't fit the infectious disease mode, okay? So things like meningitis, typhoid, the woman said, yeah. That could fit that. There's some others. There's a lot of actually human infectious diseases that where humans are actually the reservoir. Okay, staph infections, humans are reservoirs for that as well. So, okay, that's A. I need to get to B, C, and D. All right, B, a single pathogen may cause more than one disease. What's an example of that?
HPV causing multiple different kinds of cancer.
Okay, they cause cervical cancer. They cause other kinds of cancer.
They cause esophageal cancer.
Oh, okay, all right. Anybody else example of that? Yeah. Shingles. Yeah. So you have the chickenpox. I forget what that's called. Herpes, varicella zoster virus or something causes chickenpox and causes shingles later, right? Streptococcus pyogenes causes strep throat, causes flesh-eating disease, can cause, I can't remember the other one. There's like 3 or 4 different things it can cause. But that B is certainly true. A is true, B is true, C. Some pathogens may linger in the body for years. If the disease occurs, there's your HIV. Okay, HIV is one of those. Which one? Clostridium difficile. That wouldn't necessarily be one of those occur. That occurs when you have an upset to the normal microbes in your gut. When those get upset, then that Clostridium difficile will proliferate, because it's usually held in check by your normal microbes, and when they kind of go away, you're on antibiotics or something maybe, then it'll pop up, and then that's when it causes problems. Okay. Multiple pathogens can cause the same disease. Anybody have an example of that one? Let me see if somebody else puts their hand up. Anybody? What are you going to say?
Different strains of influenza can give very similar symptoms.
Yes, true. I'm thinking also of things like pneumonia, right? Pneumonia can be caused by various bacteria, can be caused by viruses, can be caused even by, I think, certain fungi. So your example as well. So basically A, B, C, and D are all true. None of these are false statements. Okay. So why do I mention this at all? Because of Koch's postulates. Okay. And don't take it to mean, though, that this is invalid, because there are these exceptions to it. Okay. But just real quickly, so what is this? And so it's a way to establish cause and effect. Is a particular microbe causing the disease? So it's based, of course, of having an animal model to do this. And you find that in all cases of disease, microbe is not found in healthy individuals. But we know there's an exception to that, because that can happen. Okay? But regardless, we do isolate that pathogen. Okay. And then we can grow it up in pure culture, and then we can take that pure culture and inject a healthy animal, and it becomes sick. Okay, and then of course we can re-isolate that same organ. So this matches what you see here and here. Right? So it has kind of a built-in repetition to make sure that you are indeed isolating the same microbe each time and that you're seeing the same disease symptoms. Okay, so the model itself is still valid. In our country, it's the CDC, Center for Disease Control in Atlanta, that they follow this model. But of course, we have knowledge, okay, of knowing about carriers that you can have healthy people that carry the pathogen. That the one disease, one pathogen doesn't necessarily apply in all cases, okay? Or even one pathogen, one disease. You can have multiple pathogens causing disease. Culturability of pathogen. He was lucky that he could culture, the diseases that he looked at, he could actually culture the pathogen. That's not always the case. Most of the microbes on planet Earth have been cultured at all. Most of us out there, we can't culture them. We don't know what they need to grow. And that's true for pathogens as well. We still haven't been able to grow in the lab the bacterium that causes syphilis. And that bug's been known for 120 years. We have yet to culture it in the lab, right? And the viruses, of course. Nothing was known in his time about viruses. If he had picked HIV as his first disease, he would have been lost, okay? No way to cultivate a virus at that time, okay? So like with a lot of things in science, sometimes it's luck, sometimes it's just hard work. But the point here is it's still valid, but we just kind of do these steps nowadays with the knowledge that we gained, right? And are aware of these exceptions, okay? Because they still do this process, okay?
So
Yeah.
If it does fall Cox isolate,
Mhm.
almost certainly it's a disease.
Yeah.
If it doesn't fall Cox
Mhm.
process, it may or may not be a disease.
Right, or it could be, but it's going to take a lot more work to figure it out, right? So, but because we know about things like viruses and whatnot, it's not going to stop us from. We just have to figure it out. It may be true, but it may not be true. But we have a lot more at our arsenal to figure these things out, research-wise, than we did 100 years ago. And just modify. If things aren't going as you would expect using cooked process, you go, okay, well, maybe it's a virus, or maybe it's this or that, and so you kind of adjust. So, obviously, once it was figured out, that microbes can be the cause of infectious diseases, it's okay, how do we kill them, right? So then, of course, the focus is on let's get rid of these things that are causing suffering in humans. And the vaccination comes into play. And vaccination, the basis of immunity. So here we see a human, and we are putting some microbes into that human, and your body has ways of fighting this infection, okay? And so one of the primary ways, here's our pathogen. What are the things you can produce to attack that pathogen? One of the main things. They look like this, right? So antibodies your body produces in response to. So antibodies recognize and bind to what? It's another A word. Antigens, right? So antibody antigen. So antigen is literally any kind of an agent that will induce your immune response, okay? And that can be anything on the periphery of a bacterial cell or virus, okay? There's various types of proteins and glycoproteins, etc., structures that your body can see and recognize and produce antibodies to. And the basis of vaccination is really about introducing an agent, okay, the antigen to your body, either in the form of a whole cell or virus that's been attenuated. Attenuated means to limit it or to render it non-disease causing. And that's what attenuated vaccines are those that are like that. They inactivate the agent, but are enough to induce an immune response. And that's your immune system that reacts to it and produces antibodies. Okay? And that's what vaccination is all about. Okay? And various Jenner and others developed these things. And I think that's probably a good place to stop, folks. So thanks for hanging in. Have a good weekend. 3-day weekend. And see you next Wednesday. And look at your picker points.